The building of protein is very essential to the functioning of an organism. In this protein building task, DNA acts as a master blueprint containing all genetic information about the protein to be created. The genetic information from DNA is transferred to messenger RNA or mRNA through a process known as transcription. Now let's learn how transcription takes place in prokaryotes such as bacteria. The prokaryotic cell does not have separations in terms of cytoplasm and nucleus. Therefore, both transcription and translation processes involved in protein synthesis can take place inside the prokaryotic cell at the same time. In prokaryotes such as bacteria, only one DNA-dependent RNA polymerase enzyme catalyzes the transcription of all types of RNA. There are three types of RNAs involved in the process of protein synthesis: messenger RNA or mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA. The mRNA carries the coding information to the sites of protein synthesis. It helps to put together amino acids to make protein. The tRNA carries each amino acid to the ribosome according to the coded message in the mRNA, while the rRNA provides a mechanism to decode mRNA into amino acids. Transcription, the first process in protein synthesis, occurs in three stages: initiation, chain elongation, and termination. In the first stage, RNA polymerase, along with the initiation factor denoted as sigma, binds to the promoter sequence in DNA and initiates transcription. This helps in the opening of the DNA helix and separates the two DNA strands. Now, the second stage of transcription, called elongation, begins. In this stage, the RNA polymerase builds a strand of RNA using one of the DNA strands as a template. RNA polymerase uses nucleoside triphosphates as substrates and polymerizes using the law of complementarity. This RNA chain growth takes place in 5' to 3' direction. After chain elongation commences, the sigma factor dissociates from the RNA polymerase and can be reused. The final stage of transcription is termination, in which the polymerase, along with termination factor represented by Rho, reaches the terminator region, and newly created mRNA fall off along with the enzyme. This marks the end of prokaryotic transcription. The stages of transcription in eukaryotes are similar to that of prokaryotic transcription. However, there are some differences as well. Let's understand them in detail. Unlike prokaryotic transcription, which occurs in cytoplasm transcription, in eukaryotes, transcription occurs in the nucleus. There is only one RNA polymerase in prokaryotic transcription. However, eukaryotic transcription involves at least 3 RNA polymerases. The RNA polymerase 1 transcribes various rRNAs. On the other hand, RNA polymerase 2 transcribes the precursor of mRNA, known as heterogeneous nuclear RNA or hnRNA. RNA polymerase III is responsible for the transcription of tRNA, 5' subunit ribosomal RNA or 5' RNA, and small nuclear RNA or snRNA. The processing of hnRNA marks another major difference between prokaryotic and eukaryotic transcription. The mRNA in prokaryotes does not require any processing. However, in eukaryotes, the hnRNA is inactive. Since it has a split gene arrangement with exons and introns, and hence it has to undergo processes such as capping, splicing, and tailing. The first process is capping, which involves an unusual nucleotide called methylguanosine triphosphate being added to the 5' end of the hnRNA. Capping helps mRNA to bind with small ribosomal subunits during protein synthesis. The next process is splicing, in which the introns are eliminated and exons are joined in a specific order. Finally, in the tailing process, about 200 to 300 adenylate residues are added to the 3' end of the hnRNA in a template-independent manner, giving rise to a poly-A tail. This process is also known as polyadenylation. After these processes, the hnRNA becomes mRNA and the transcription process ends. The mRNA can now be transported out of the nucleus and further used for the process of protein synthesis during translation.